there's another enzyme besides PFK1. Remember we were saying, oh, it's PFK1. Well, here's PFK2. You knew it was coming. Right? PFK2 makes fructose 2,6-bisphosphate instead of fructose 1,6-bisphosphate. So it's a different enzyme. Well, the mechanism is similar, but it's a different enzyme. And then there's another phosphatase that takes it back off. So you're going back and forth. So the source of it is F6P. Right? So, and these two activities, by the way, are also part of the same protein. Right? The same covalent protein actually has both active sites on it. And one of the active sites is active and the other one is not at any time, any particular time. So that's kind of an interesting twist on it. So when PFK2 is active and this guy is inactive, then there's more F26BP, right? Because PFK2 is active, so there's more of this. And if there's more of this, then glycolysis is going forward by virtue of its effects on PFK1. Right? And the other way around, glycolysis inhibited, gluconeogenesis is stimulated. So these are the two states, right? And now here, coming in from the sides, are hormonal control of those states, right? So here's insulin through some pathway, which you probably learned something about last term, activating a phosphatase, which takes the phosphate off of this protein, which results in one of these subunits being active. Which one? Well, the one that stimulates glycolysis. Right? So more glycolysis in response to insulin. Insulin signals high blood glucose. We want more glycolysis if there's a lot of insulin around. Right? And similarly here, it goes, it's glucagon, we already talked about what that does. So glucagon is going to stimulate this reaction so that this guy goes down, right? And F26BP goes down, then it's not around to inhibit gluconeogenesis, so gluconeogenesis will be stimulated while the lack of F26BP here will knock out the glycolysis, which is what you want if you have low blood sugar. That make sense? So it's straightforward, but it's a little complicated. So it's worth kind of writing it down and just thinking it through. It totally makes sense. It's not like, oh, this is a big trick in here somewhere. It's just that the mechanism is a little bit complicated. But you know, that's kind of how cells work. I only have time for a couple questions. Yeah.
So it's one of those exact questions. So should we know all the steps and all the enzymes, or should we focus more on learning the basic principles?
Yeah, here for regulation, you should certainly focus on the principles. I will ask some specifics about it, but we'll talk about that as we go on for the review. But you should, this kind of stuff, it's good to know this, I think, pretty well. Yeah. Okay, one more question. No. Okay, see you guys on Friday.